Welcome everyone to chapter 23. This is the digestive system. In this chapter, we're going to look at the parts of the digestive system and their functions, as well as the different types of digestion. Now, unfortunately, we won't go into much detail of how different diets affect the body, but instead we will be more going over how macromolecules are used by the body to create energy. If you are interested in specific diets and their effect on the body, I would suggest maybe a diet and nutrition specific class. So two groups of organs make up the digestive system. These are the GI tract and then the accessory organs. So GI tract stands for gastrointestinal tract. This is a long continuous tube that starts at the mouth and it will continue all the way to the anus. The accessory structures are organs or glands that are attached to the gastrointestinal tract. So like I said, the GI tract is going to be this long continuous tube, and it goes through the thoracic and abdominal cavities and extends from the mouth to the anus. Organs of the GI tract, these are going to include the mouth, most of the pharynx, and then the esophagus, the stomach, small intestine, large intestine, and the rectum. The accessory digestive organs are those that are not part of the GI tract, but they do contribute to digestion and processing of food. So these are going to be things like the teeth, the tongue, salivary glands, the liver, gallbladder, and pancreas. Overall, the digestive system performs 6 basic processes. Our first function or first process is ingestion. So taking in foods and liquids into the mouth. So we know that as eating and drinking. So that's our first function. Our second function is secretion. So each day, the walls of the gastrointestinal tract and the accessory organs secrete about 7 liters of water, acids, buffers, and even enzymes into the lumen of the gastrointestinal tract. Our third function of the digestive system is mixing and propulsion. So we get alternating contractions and relaxations of smooth muscle in the walls of the GI tract, will mix the food and secretions, and also move material along the length towards the anus. So the digestive system is going to mix the food that you eat and mix it with the enzymes and acids and everything. And it's also going to move that food along the entire length of the gastrointestinal tract. Our fourth function is digestion. So these are going to be both mechanical and chemical processes that break down the ingested food into small molecules. The fifth function is absorption. So this is the bringing in the ingested and secreted fluid, ions, and all the products of digestion into the epithelial cells lining the lumen of the GI tract and moving it into the blood and the lymph. Essentially, we're going to take all those small molecules that were mechanically and chemically broken down, and we are going to absorb them from the GI tract into the blood and the lymph. So that's our fifth function of the digestive system, absorption. And then our sixth function is defecation. Wastes, indigestible substances, bacteria, and digested material that wasn't absorbed in their journey are going to leave the body through the anus. The entirety of the wall of the gastrointestinal tract has the same basic four-layered arrangement of tissues. Now, the four layers of the tract, from deep to superficial, are the mucosa, the submucosa, the muscularis, and the serosa in most parts. In other parts, it's called the adventitia. We'll talk about the differences between the two. Okay, so first off, we'll talk about the mucosal layer. This is the inner lining of the gastrointestinal tract, and essentially it's a mucous membrane with endocrine cells that secrete hormones, mucus, and other secretions. So this is going to be the layer of the GI tract just adjacent to the lumen, or the inside of the tube. So that's that most interior layer of the tube. Next, we have the submucosa layer. It's kind of this white layer here. Now, this consists of areolar connective tissue, and it's going to bind the mucosal layer to the muscularis layer. Now, in this layer, there's going to be a lot of blood vessels and lymphatic vessels that receive the absorbed food molecules. Now, next is the muscularis layer. So the muscularis at the beginning of the gastrointestinal tract, the mouth, the pharynx, and then the superior parts of the esophagus, is going to contain skeletal muscle that produces voluntary swallowing. So this is just going to be at the beginning of the GI tract. This is going to be skeletal muscle. So again, that's just going to be like the mouth, the pharynx, and then the superior part of the esophagus. Now, throughout the rest of the GI tract, though, this muscularis layer consists of smooth muscle, so that it's a non-voluntary muscle. You can't really control how quickly the food flows through your small intestine or your large intestine, for example. Because it's that smooth muscle, it's involuntary in that muscularis layer. Now we have the most superficial layer. So the portions of the GI tract that are suspended in the abdominal cavity have a superficial layer called the serosa. So these are ones that are suspended in the abdominal cavity. Then your superficial layer is called the serosa, and this is a serous membrane. Now in the esophagus, we don't have a serosa. Instead, there's only a single layer of areolar tissue called the adventitia, and this forms the superficial layer of the gastrointestinal tract. So this is only going to be in the esophagus. This is in the non-suspended portion of the GI tract. Throughout the whole rest of the GI tract, that's all suspended. It is a serosa. Now, the GI tract is regulated by an intrinsic set of nerves known as the enteric nervous system. And we also have an extrinsic set of nerves that are part of the autonomic nervous system. So this is going to be intrinsic or inside, and this is extrinsic or originating from outside of the gut. So recall that the brain of the gut is the enteric nervous system, and this is going to extend along the entire GI tract. Now, the neurons of the enteric nervous system are arranged into two plexuses, the submucosal plexus and the myenteric plexus. These plexuses of the ENS, the enteric nervous system, are going to consist of the normal neurons that we'd think of. These are going to be motor neurons, interneurons, and sensory neurons. Now, even though the neurons of the ENS can function independently, they really are subject to regulation by the neurons of the autonomic nervous system. So these are going to be usually regulated by the neurons of the autonomic nervous system. Now, the vagus nerve, remember this is the 10th cranial nerve. This is going to supply the parasympathetic fibers to most parts of the GI tract. And in general, the parasympathetic portion is going to increase GI secretion and motility and increase the activity of the ENS neurons. So increased GI secretion, increased GI is just gastrointestinal motility. So essentially how quickly food moves through the gastrointestinal tract. And the vagus nerve will also increase the enteric nervous system neuronal activity. So this is what the vagus nerve does, or the vagus nerve activates. Now, on the other hand, the sympathetic nerves that supply the GI tract are going to have kind of the opposite effect. So the sympathetic nerves are going to have a decrease in gastrointestinal secretion and motility. And really they do this by inhibiting the neurons of the ENS. This slide is just kind of reiterating that the enteric nervous system is regulated by the autonomic nervous system, by that vagus nerve of the parasympathetic fibers, and then the sympathetic fibers as well. So these reflex pathways by the autonomic nervous system are really going to regulate that gastrointestinal secretion and motility. Now let's talk about the membranes of the gastrointestinal tract. So the peritoneum, this is the largest serous membrane of the body. Now it consists of a layer of simple squamous epithelium with this underlying supporting layer of areolar connective tissue. Now the peritoneum is divided into a parietal peritoneum, which lines the wall of the abdominal cavity, and then the visceral peritoneum, which covers some of the organs in the cavity and their serosa. This is going to be very similar to all the other serous membranes that we've talked about so far, having those two layers, parietal and visceral layer, one that covers the abdominal wall and the other that covers the organ. Now, unlike the pericardium and the pleura, which will very smoothly cover the heart and the lungs, the peritoneum contains these large folds which will kind of weave between the viscera. Now there are 5 major peritoneal folds. So these are the greater omentum, so these are the major peritoneal folds, greater omentum, the falciform ligament, the lesser omentum, then there's the mesentery and the mesocolon. So we'll just briefly discuss each of these. So first, we can see here we have the greater omentum. So this is the largest peritoneal fold. It's going to drape over the transverse colon, and it's also going to drape over all of the coils of the small intestine, kind of like this fatty apron. So you can kind of think of this as kind of a apron kind of made of fat, essentially, is what the greater omentum is. Now this is going to be the layer or the fold that really contributes to the beer belly look of some overweight individuals. So it gives us this big, large belly if there's a lot of adipose tissue added to the greater omentum. Then we have the falciform ligament. This attaches the liver to the abdominal wall and the diaphragm. So liver to the anterior abdominal wall. This is where it attaches to. Now, one really important note is that the liver is the only digestive organ that attaches to the anterior abdominal wall. And this is by the falciform ligament. Again, the liver is the only digestive organ that attaches to the anterior abdominal wall. Next is the lesser omentum over here. This arises as an anterior fold in the serosa of the stomach and the duodenum. And really it connects the stomach and the duodenum to the liver. This is what the lesser omentum does. Connects the stomach and the duodenum of the small intestine to the liver. Then we have this fan-shaped fold of the peritoneum, which is called the mesentery. Now this is going to bind the jejunum of the small intestine and also the ileum of the small intestine and it binds these two parts of the small intestine to the posterior abdominal wall. So the mesentery is going to bind, or kind of connect, the jejunum and the ileum to the posterior abdominal wall. Now lastly, there are two separate folds of the peritoneum called the mesocolon. I actually don't have a picture of this one here, but this will bind the transverse colon and the sigmoid colon of the large intestine to the posterior abdominal wall. So this will bind the transverse colon and also the sigmoid colon to the posterior abdominal wall. The mouth, which is also referred to as the oral cavity, is formed by the cheeks on the sides, and then the hard and soft palate on the top, and then the tongue on the bottom. Now the palate is a wall or septum that separates the oral cavity from the nasal cavity, and it forms the roof of the mouth. Now this is a really important structure that makes it possible to chew but also breathe through your nose at the same time. The hard palate, this is the anterior portion of the roof of the mouth, and it's formed by the maxillae and palatine bones. Oops, maxillae. So this is going to be kind of the bony palate, and it's covered in mucous membrane. So this is going to form the bony partition between the oral and nasal cavities. Then we have the soft palate, and this forms the posterior portion of the roof of the mouth. And it is an arch-shaped muscular partition. And this is between the oropharynx and the nasopharynx. And it's also going to be lined with mucous membrane. Salivary gland is a gland that releases a secretion called saliva into the oral cavity. Now, ordinarily, there's just enough saliva that's secreted to keep the mucous membranes of the mouth and pharynx moist, as well as to cleanse the mouth and the teeth. Now, most of the saliva is secreted by 3 pairs of major salivary glands. Now, these lie beyond the oral mucosa, just outside the oral mucosa, and they have ducts that lead into the oral cavity. Now these 3 pairs of major salivary glands, these are going to be the parotid gland, the submandibular gland, and then the sublingual. So they're going to be found in pairs, 1 on each side of the oral cavity. The parotid glands, these are located just inferior and anterior to the ears. So it's going to be this gland right here. And it's located between the skin and the masseter muscle. So here's that masseter muscle here. Now, each of the parotid glands secretes their saliva, the saliva that they produce, into the oral cavity via the parotid duct. This duct is going to open into the mouth near the second maxillary molar tooth. Next are the submandibular glands, which is this gland down here. These are found kind of on the floor of the mouth, and they're found medial and partially inferior to the body of the mandible. And then the submandibular duct is going to be right here, or labeled right there. It's a very small duct. It's kind of hidden in this picture. It's going to be right here. Kind of right there is where that duct is. And then lastly are the paired sublingual glands. These glands right here. Now these are found beneath the tongue, and they are superior to the submandibular glands. The tongue is an accessory digestive organ composed of skeletal muscle covered with a mucous membrane. Now, together with the associated muscles, it forms the floor of the oral cavity. Now, there are kind of two different types of muscles that are associated with the tongue. These are the extrinsic muscles of the tongue, and the intrinsic muscles of the tongue. So the extrinsic muscles of the tongue, these are going to originate outside the tongue. And these muscles attach bones to the connective tissues inside the tongue. And they're able to move the tongue from side to side and in and out in order to maneuver food for chewing, to shape the food into a rounded mass, and also to force food to the back of the mouth for swallowing. So those are all the things that the extrinsic muscles of the tongue help the tongue to do. Move the tongue from side to side and in and out of the mouth, to maneuver food for chewing, to shape the food into a rounded mass, and then to force the food to the back of the mouth for swallowing. Now the intrinsic muscles of the tongue, these are going to originate inside the tongue. And they will insert into connective tissue within the tongue. Now these muscles are a little bit different than the extrinsic muscles, as the intrinsic muscles will alter the shape and size of the tongue, mainly to be used for speech and swallowing. Next, moving on to the teeth. Now the teeth are accessory digestive organs that are located within sockets of the alveolar processes of the mandible and the maxillae, so the upper and lower jaws. Now a typical tooth has three major external regions. These regions are going to be the crown, the root, and the neck. So the crown, this is going to be the visible portion. It's above the level of the gums. So here we have the gingiva, which is also known as the gums. And so the crown is found above the gum line. Now embedded in the socket are going to be 1 to 3 roots. So these are the parts that are below the gum line into the socket. And then the neck is just the constricted junction of the crown and the root right near the gum line. Now a couple different parts of a tooth. We have dentin. So this forms the majority of the tooth, and it's calcified connective tissue here, and essentially dentin is going to give the tooth its basic shape and rigidity. Now the dentin of the crown is covered by enamel. This consists primarily of calcium phosphate and calcium carbonates, so we could just say some calcium salts. And enamel is actually going to be the hardest substance in your body. It's going to be harder than bone, and this is because it has an even higher content of calcium salts. Now humans have 2 dentitions, or also known as sets of teeth. So essentially we have a deciduous set of teeth, or deciduous dentition, and a permanent dentition. So these are both labeled here, deciduous dentition and then the permanent dentition. So our deciduous teeth, which are also kind of known as baby teeth, these usually begin to erupt at about 6 months of age. So here we can see our central incisor usually comes out about 6 to 8 months. And approximately 2 teeth appear for every month after, until 20 teeth are all present. So we have 20 deciduous teeth. So all of these teeth kind of have when they erupt, all the way up to 32 months for our second molars there. Now, all of our deciduous teeth are lost. Generally, these are going to be lost between the ages of 6 to 12 years old. And all these teeth are going to be replaced with permanent teeth, or our adult teeth. Now, the permanent dentition contains 32 teeth, and these will erupt between the ages of about 6 to 7 years old, with our first molar here at about 6 years old, and they will continue to erupt between 6 to adulthood. Usually here are our wisdom teeth. If you don't have your wisdom teeth taken out, generally they will erupt from your gum line between about 17 to 21 years old. Now there are 2 types of digestion we're going to talk about throughout this chapter. These are mechanical digestion and chemical digestion. So in the mouth, mechanical digestion essentially results from chewing, which the scientific word for chewing is mastication. So mastication means chewing. And this is where the food is manipulated by the tongue. It is kind of moved around or crushed by the teeth, and it is mixed with saliva. Now, as a result, the food is reduced to a really soft and flexible, easily swallowed mass, which is called a bolus. So essentially the bolus is going to be this massive food that's been crushed and chopped with by your teeth and mixed with saliva and everything, this mushy, massive food that you can easily swallow. So in your mouth, this is called a bolus. Now there's also some chemical digestion that happens in the mouth as well. Now there are two enzymes, salivary amylase, and then there's also going to be lingual lipase, which are going to be two important enzymes in your mouth. Salivary amylase and lingual lipase, these are both going to contribute to chemical digestion in the mouth. So salivary amylase is secreted by the salivary glands. This essentially initiates the breakdown of starch. So it just kind of starts that starch breakdown, and starch will continue to be broken down as it moves through your digestive system. Now, saliva also contains lingual lipase, so it's going to be secreted by the salivary glands as well. And this is mainly going to be secreted by the lingual glands in the tongue. But this one is going to break down triglycerides. So it's going to break down those fats and oils. So remember, triglycerides, these are going to be fats and oils. Again, this is just going to start or initiate that digestion of triglycerides, and triglycerides are going to be further continued to be chemically digested as they move down your digestive system tract or your gastrointestinal tract. So here is a great table that shows the digestive activities in the mouth. So kind of get all these structures here, cheeks and lips, salivary glands, the tongue, and your teeth. Essentially, the activity of all of them, what all of them do, and then the result as well. So good studying table here. Now, when food is first swallowed, it passes from the mouth into the pharynx. We've already kind of talked about the pharynx in the respiratory tract, but again, the pharynx is going to be this large structure, only about a couple 5 inches long, I guess, moving from the internal nares in the nose down to the esophagus and larynx on the posterior side. So again, here we can see these full regions of the pharynx here. But this is where food is going to pass from the oral cavity down into the pharynx here. Now the pharynx is composed of skeletal muscle and lined by mucous membrane, and we know that it's divided into these three parts: the nasopharynx, oropharynx, and laryngopharynx. We know that the nasopharynx functions only in respirations, so it's only going to be part of the respiratory tract, but both the oropharynx and laryngopharynx have digestive functions as well as respiratory functions. So swallowed food is going to pass from the mouth or from the oral cavity into the oropharynx and then the laryngopharynx. Now, the muscular contractions of these areas will help propel the food into the esophagus and then into the stomach. The esophagus, and this is a collapsible muscular tube that lies posterior to the trachea, and it begins at the inferior end of the laryngopharynx, and it passes through the inferior aspect of the neck, and then enters the mediastinum anterior to the vertebral column. Now, near the stomach, the mucosa of the esophagus also contains mucus glands, but that's only going to be near the stomach. Superior to that portion, the esophagus doesn't have mucus glands. Now, remember when we were talking about the different layers of the gastrointestinal tract, we talked about how the esophagus is a little bit different from the rest of the GI tract. So the most superficial layer of the esophagus is known as the adventitia, and this is as opposed to the serosa, as in the stomach and the intestines. And this is called the adventitia because the areolar connective tissue of this layer is not covered by mesothelium, and also because the connective tissue merges with the connective tissue of surrounding structures of the mediastinum through which it passes. In short, or what that also means, is that the adventitia attaches the esophagus to the surrounding structures. That's very much unlike the serosa of the stomach and the small intestine, which attaches the stomach and small intestine to the walls, the thorax walls, or the abdominal walls. Okay, so adventitia will attach the esophagus to surrounding structures, but the serosa of the stomach and small intestine does not attach the stomach and small intestine to surrounding structures. The movement of food from the mouth into the stomach is achieved by this act of swallowing, also known as deglutition. Up here at the top, deglutition means to swallow. Now, deglutition is facilitated by the secretion of saliva and mucus, and it's involved in the mouth, the pharynx, and the esophagus. Now, there are kind of three steps to deglutition, or three phases. These are going to be, or three stages, I guess we'll call them. The voluntary stage, the pharyngeal stage, and then the esophageal stage. So swallowing starts when the bolus is forced to the back of the oral cavity and into the oropharynx by the movement of the tongue. The tongue will move upward and backward against the palate. So these actions will constitute the voluntary stage of deglutition. Just the forcing of the bolus to the back of the oral cavity by the tongue. Now, with the passage of the bolus into the oropharynx, this is when the pharyngeal stage of deglutition begins, and this is an involuntary stage. And this pharyngeal stage of deglutition is where the passage or the bolus is going to pass through the pharynx into the esophagus. So voluntary stage is moving the bolus to the back of the oral cavity with the tongue, which is a voluntary action. And then the pharyngeal stage is just that movement of the bolus from the oral pharynx, or I guess through the whole pharynx, into the esophagus. And this is an involuntary stage. Then lastly is the esophageal stage of swallowing. And this stage begins once the bolus is in the esophagus. So we have the esophagus labeled here. Now, during this stage, this is where peristalsis occurs. So peristalsis is this progression of coordinated contractions and relaxations of the circular muscles and longitudinal muscles of the muscularis, which are going to push the bolus forward. So when the longitudinal muscles contract, so if we have like a relaxed longitudinal muscle, when it contracts, it gets shorter this way. And so we're going to essentially be shortening the tube when the longitudinal muscles contract. So we have kind of a tube like this of longitudinal muscles, and there's a bolus in here. When the longitudinal muscles contract, they just essentially get shorter, so that the bolus is kind of further along in the pathway. When the circular muscles contract, they're going to have this wave of contractions squeezing it down. Kind of wave down, squeezing it further and further and further as the bolus continues to travel down. Now, peristalsis does occur in other tubular structures as well, including other parts of the GI tract, in the ureters as well, and then even in the bile ducts and the uterine tubes. We also have this peristalsis occurring by circular and longitudinal layers of the muscularis layer. Now, the esophageal stage of deglutition will end when the bolus makes it to the stomach. So in this table, we can see the digestive activities of the pharynx and esophagus. So in the pharynx, we get that pharyngeal stage of deglutition, and then we have our results on the right here. And then we have the esophagus, and then the activities at the esophagus as well. The stomach is the J-shaped enlargement of the gastrointestinal tract, and it's found directly inferior to the diaphragm in the abdomen, so directly below the diaphragm. Now, there are 4 main regions of the stomach. First is the cardia. This is the part of the stomach surrounding the opening of the esophagus into the stomach. Next is the fundus. This is the rounded portion superior to and to the left of the cardia. Kind of this big dome-shaped area here, that's the fundus. Then we have the body. This is the large central portion of the stomach. And then kind of our fourth section of the stomach, we call the pyloric part. And the pyloric part has three regions itself. So the pyloric part is made up of the pyloric antrum, the pyloric canal, and the pylorus. And then the pylorus will kind of empty the stomach into the duodenum. So kind of this section kind of right here is going to be that fourth pyloric part area, made up of a couple different sections. And there's a concave border of the stomach, which we call the lesser curvature, and then a large convex lateral border that we call the greater curvature. Now, in general, there are four functions of the stomach. So our first one has to do with mixing. So it mixes everything that you ingest along with saliva and gastric juice in order to form chyme. Chyme is just essentially the pulpy, partially digested acidic food. So from the pharynx down the esophagus, your partially digested food is called a bolus. After the stomach, we call it chyme. Our second function of the stomach is that it serves as a reservoir for food. The third function is that it secretes gastric juice, which contains hydrochloric acid. So it kills and denatures everything. So this word denature, this means that proteins unravel and become useless. So denaturing is the unraveling of proteins, making them useless. Because proteins, they kind of twist and turn and kind of make this globular mass, but it's a very specific globular mass, and it gets its function from the specific orientation that that the amino acids are kind of oriented in. So when you kind of just pull apart that globular mass, it completely loses all of its functioning. So that's what denaturing is. Now, the fourth function of the stomach is that it also secretes gastrin into the blood. Now, gastrin is a hormone that stimulates the secretion of gastric juice. So this is going to stimulate the secretion of gastric juice. Now, the stomach wall is composed of the same basic layers that the rest of the gastrointestinal tract is made up of, with a couple modifications. So we have the most superficial layer being the serosa, then the muscularis layer with a circular layer of muscle and an oblique layer of muscle, kind of diagonally arranged. Then we have the submucosal layer with all the blood vessels and the lymphatic vessels in that layer. And then the mucosa layer. Now the mucosa layer is going to be kind of the most modified of all the layers between the rest of the gastrointestinal tract here. So we see these really long kind of finger-like projections here, but these are all kind of like pits. So they're kind of circular, and it makes these long divots here that are pits. Along the lining of these pits, we have lots of different cells, mucous neck cells, parietal cells, chief cells, G cells, and then there's also going to be glands down here as well. And we'll talk about all of these different cells within the mucosal layer of the stomach as well. The surface of the mucosa is a layer of simple columnar epithelial cells, which are called surface mucus cells. These epithelial cells will extend down into the lamina propria, where they form columns of secretory cells, which are called gastric glands. So down here, down in the pit, these are going to be the glands. Now, several of these gastric glands will open into the bottom of narrow channels, and it's going to be these channels that are called the gastric pits. So secretions from several gastric glands will flow into each gastric pit and then into the lumen of the stomach. So the gastric glands contain 3 types of exocrine gland cells. So these exocrine glands, what exocrine? So these exocrine gland cells are going to be the mucous neck cell, parietal cells, and chief cells. So those are the exocrine gland cells within the gastric glands. So these are going to secrete their products into the stomach lumen. So they'll secrete it into the glands. Those secretions will go up the gastric pit, out into the lumen of the stomach. So mucous neck cells, these guys secrete mucus. Parietal cells, these produce what's called an intrinsic factor. This is needed for the absorption of B12. So needed to absorb B12. And then these parietal cells will also secrete hydrochloric acid too. And then the chief cells. These secrete pepsinogen, which we will talk about yet. And then they also secrete gastric lipase. So gastric lipase is very much like lingual lipase. They're both lipase enzymes that break down triglycerides. They're just found in different locations. So gastric lipase is found within the stomach. Lingual lipase is found in the mouth. Now it's going to be the secretions of the mucus, parietal, and chief cells that form gastric juice. Now you might see that there's one more cell here that we haven't really talked about yet. So in addition, the gastric glands include a type of enteroendocrine cell. So this is the G cell. So the G cell will secrete the hormone gastrin into the bloodstream, which essentially is going to stimulate all three of the other cells. Because remember, gastrin is going to stimulate the secretion of gastric juice. And gastric juice is made from all three of these exocrine gland cells. So the G cell is going to secrete gastrin, which stimulates all the three other gland cells. The enteroendocrine, this word just means it is a gastrointestinal tract endocrine cell. So that it secretes its products into the interstitial fluid, and then it will get absorbed into the bloodstream. So here we have a parietal cell. So parietal cells, remember, like I said, they secrete hydrochloric acid. It's one of the secretions that they produce. So parietal cells will actually secrete hydrogen ions and chloride ions separately into the stomach lumen. But the net effect of that secretion is going to be hydrochloric acid. Now, as we quickly go over all these chemical reactions that occur in the parietal cell, let's first quickly take a look at the key down here. So just so we know what all of these symbols and everything mean. So this little red circle with the arrow through it, this is a proton pump. Ca stands for carbonic anhydrase. Little dotted arrow is going to be diffusion. These little channels, I guess they're slightly different colors. The top one is black. This is a potassium ion channel. Bottom one looks kind of purple. This is the chloride ion channel. And then our last little thing here, this brown box with arrows through it, is a bicarbonate chloride antiporter. So we already kind of know the end product that parietal cells send out, but where does this start? So it starts here with water and carbon dioxide. Now we have this carbonic anhydrase that will convert water and carbon dioxide into carbonic acid. Carbonic acid will dissociate into hydrogen ions and bicarbonate. And that hydrogen ion will come all the way over here, where a proton pump will actively transport hydrogen into the lumen, while at the same time bringing potassium in. So this is a hydrogen potassium ATPase. So it will pump hydrogen out and at the same time pump potassium ions into the cell. Now this pumps potassium in, but the cell doesn't really actually need potassium. It's just kind of how the cell works. So we can kind of see this little line right here. This is going to be a potassium ion channel. So potassium is going to diffuse out back into the lumen through these potassium channels in the apical membrane. Chloride, which is pumped in when bicarbonate is pumped out at the basolateral membrane here. As this potassium is being pumped out at the top, this chloride is also going to be pumped out into the stomach lumen as well. So we get these chloride and potassium diffusing out into the lumen through these chloride and potassium channels in the apical membrane. Now this bicarbonate from this reaction here is going to be pumped out with that bicarbonate chloride antiporter right here. And it's going to pump out that bicarbonate, remember, which is a buffer molecule. It's going to pump it into the blood capillary in the lamina propria. So pump it out there. So that's essentially how we get the hydrogen and chloride ions being pumped out into the stomach lumen from the parietal cell. That carbonic anhydrase is going to catalyze the formation of carbonic acid from water and carbon dioxide. And that's because this carbonic anhydrase is going to be very, very plentiful in these parietal cells. Parietal cells have a ton of carbonic anhydrase, so they can pretty much always convert water and carbon dioxide into carbonic acid. Carbonic acid very quickly dissociates into hydrogen and bicarbonate, and it provides this ready source of hydrogen for the proton pumps. But it also generates bicarbonate ions that can exit the cell in exchange for chloride. And this is because of these bicarbonate chloride antiporters in the basolateral membrane. So this is another very important aspect of this process because we need those chloride ions to be pumped in from the interstitial fluid so that they can go out to the lumen. Now on the right here, we have this other picture, pretty much the same parietal cell as well, but it shows that this hydrochloride secretion can be stimulated by several sources. So these sources on the right, on the basolateral membrane, are all going to increase hydrochloride secretion by the parietal cell. So acetylcholine, gastrin, and histamine all cause parietal cells to secrete more hydrochloric acid. So here we have a really nice diagram for showing what cells do within the stomach mucosal layer. So here we have the mucosal layer, we have surface mucus cells and also the mucous neck cells, all their activity and the result. Parietal cells, chief cells, and G cells are all on this diagram. Here it also shows the functions of the muscularis layer. the muscular layer of the stomach, and then also the pyloric sphincter. Now, this pyloric sphincter is a ring of muscle that serves to close the opening of the stomach to the small intestine. So it's going to open to allow the passage of chyme into the duodenum of the stomach, or of the small intestine, sorry, from the stomach into the small intestine. Now, from the stomach, chyme passes into the small intestine. Now, chemical digestion in the small intestine depends on the activities of the pancreas, the liver, and the gallbladder. So because of that, we're first going to talk about these accessory organs and their contribution to digestion in the small intestine. So the first one we have here is the pancreas. Now, this is a gland that lies posterior to the greater curvature of the stomach. The pancreas consists of a head, which is the larger portion, the body, and a tail. And it's going to produce enzymes as well as sodium bicarbonate that are brought into the duodenum. So it produces these enzymes and sodium bicarbonate that are emptied from the pancreas into the duodenum of the small intestine. Pancreatic juices are secreted by exocrine cells into a ton of small ducts that will ultimately unite to form two larger ducts. These are the pancreatic duct and the accessory duct. So this duct is over here in this picture. So these are going to be the two main ducts exiting the pancreas. These are going to bring the secretions of the pancreas to the small intestine. So that is the pancreatic duct and the accessory duct. Now, the pancreatic duct is the larger of the two ducts, and it will join with the common bile duct from the liver and gallbladder, and will enter the duodenum as a dilated common duct known as the hepatopancreatic ampulla. The pancreas is made up of small clusters of glandular epithelial cells. About 99% of these clusters, called acini, these constitute the exocrine portion of the organ. They secrete a mixture of fluid and digestive enzymes, which is called pancreatic juice. So they're going to secrete their pancreatic juice into these ducts, which will then go to those two main ducts, like we said before, the pancreatic duct and the accessory duct. Now, the remaining 1% of clusters are called pancreatic islets, also known as islets of Langerhans. So these are also known as pancreatic islets. And these islet of Langerhans are going to constitute the endocrine portion of the pancreas. So here we have this big blown up portion or blown up pancreatic islet down here. Now, the cells of the pancreatic islet are going to secrete the hormones glucagon, insulin. Different cells secrete these different hormones. Somatostatin, and pancreatic polypeptide. And we'll discuss these specifically more when we talk about the endocrine system. Now, the liver makes bile. And then it sends that bile to the gallbladder for storage. So the gallbladder will store the bile where the liver makes the bile. Now, bile is very, very important for the emulsification of fats. Now, this term emulsify essentially means to kind of disperse a liquid together with another liquid. So when we're talking about fats, it's going to break down like one big glob of fat into much smaller portions. So we're going to greatly increase the surface area of the fat so that it can be broken down easier. So if we have like a tub of water here and we put some oil, the oil will stay in one big glob in the water. Now, if we add an emulsifier with these fats, say we put bile in this solution, it will break down that oil into lots of little droplets. And so the surface area is greatly increased in these fats, so they can actually be broken down a lot easier. So the bile is sent through the common hepatic duct from the liver, and it can go to the gallbladder through the cystic duct, and that's going to be for storage. And then it will go to the small intestine by the common bile duct. Now, histologically, the liver is composed of several components. First, there are hepatocytes. Now, these are also these little orange cells in here in this right picture. These are going to be the major functional cells of the liver, and they perform a really wide variety of metabolic, secretory, and endocrine function. Now, the hepatocytes create the bile that we just spoke about, and they secrete the bile that they produce into these little bile canaliculi. These are going to be small ducts between the hepatocytes. Now, from the bile canaliculi, the bile will pass into bile ductules and then into bile ducts. The bile ducts will merge, and eventually they're going to form the right and left hepatic ducts. Hepatic sinusoids You also would have seen those in the pictures in the last slide. These are going to be really highly permeable blood capillaries between the rows of the hepatocytes. And they receive oxygenated blood from branches of the hepatic portal vein. And these hepatic sinusoids will converge and deliver blood into a central vein. Now together, a bile duct, a branch of the hepatic artery, and a branch of the hepatic vein are referred to as a portal triad. The hepatocytes, bile duct system, and the hepatic sinusoids can be organized into anatomical and functional units in 3 different ways. So first way we have is the hepatic lobule. Now for years, this was thought to be the functional unit of the liver. In this model, each hepatic lobule is shaped like a hexagon. So we can see this hepatic lobule here, shaped like a hexagon. And there's a central vein in the center of each hepatic lobule, with 3 corners of the hexagon being a portal triad. Though this is a very specific and well-defined model, it's kind of difficult to find this specific organization found in the human liver. Now, the next functional unit we'll talk about is the portal lobule. Now, this model emphasizes the exocrine function of the liver. So that is bile secretion. So this one is going to emphasize bile secretion. Now, because of this, the bile duct of a portal triad is taken as the center of the portal lobule. So the hepatic lobule is going to be this dotted line around the hexagon, whereas the portal lobule is kind of this triangular shape where we have the bile duct of the portal triad in the center of the portal lobule. So it's going to be this triangular in shape, kind of defined as 3 imaginary straight lines that connect these 3 central veins that are closest to the portal triad. So this is the portal lobule. And then our final kind of model that we can look at is a hepatic acinus. So this is kind of the preferred structural and functional unit of the liver. So each acinus is approximately an oval mass that includes 2 neighboring hepatic lobules. Now the hepatocytes in the hepatic acinus are arranged into 3 zones. We'll actually get detail here on the left of a hepatic acinus. So this is just going to be a zoomed in version of that. So we can see here that the hepatocytes in the hepatic acinus are arranged into 3 zones around the short axis. So if we're looking at this one on the right here, the zones are going to be kind of arranged like this, and like this, and like this. So kind of see the zoomed up picture on the left here. There's really not going to be sharp boundaries between these 2 zones. It's kind of a rough estimate here. So the zone 1 cells, these are going to be closest to the branches of the portal triad. And essentially, they're going to be the first to receive incoming oxygen and nutrients, but they're also going to be the first to receive toxins coming from the incoming blood. They're going to be the first to be able to take in glucose and be able to use it, but they're also the first to be affected by the toxins. Zone 2 is a little bit further out. And zone 3 is going to consist of the hepatocytes furthest from the portal triad. Now the liver receives blood from two sources. It gets oxygenated blood from the hepatic artery, and then the hepatic portal vein also provides deoxygenated blood containing newly absorbed nutrients, drugs, and even possibly microbes and toxins from the gastrointestinal tract. So both of those blood sources are going to go through the hepatic sinusoids, to the central vein, hepatic vein, and then through the systemic circulatory system. Now every day the hepatocytes secrete about 800 to 1,000 milliliters of bile. Now bile is this kind of yellowish brownish liquid, and it has a pH of about 7.6 to 8.6, and it consists mostly of bile salts, cholesterol, and several ions. So it's a yellowish brownish liquid, mostly has bile salts, cholesterol, and ions. Now the principal pigment in bile is a pigment called bilirubin. This is derived from hemes in red blood cells, and it is secreted into bile and then broken down in the intestine. Bilirubin is a yellowish pigment. It's going to be made by the breakdown of red blood cells. Now, another one of the breakdown products made in the liver is stercobilin. So stercobilin gives feces their normal brown color. So bilirubin's more of a yellowish color, and stercobilin is more of a brown color. Now, bile is partially an excretory product and partially a digestive secretion. Bile salts, which are really going to be sodium salts and potassium salts of bile acids, these are going to play a role in emulsification. So remember, emulsification is going to be that breakdown of large lipid globules into kind of a suspension of a lot of smaller lipid globules. And these small lipid globules present a very large surface area that allows pancreatic lipase to more rapidly accomplish digestion of those triglycerides. So this emulsification increases the surface area of those lipids and allows for pancreatic lipase to quickly digest the triglycerides. Bile salts also do kind of aid in the absorption of lipids following their digestion as well. Now, in addition to secreting bile, which is needed for the absorption of dietary fats, the liver performs many other vital functions. So first is going to be carbohydrate metabolism. So this is maintaining or helps to maintain a normal blood glucose level. The liver also performs in lipid metabolism. And this is because the hepatocytes will break down fatty acids to generate ATP. And the liver also works in protein metabolism as well. So hepatocytes will work to remove the amino group from amino acids so that the amino acids can be used for ATP production. The liver will also function in processing of drugs and hormones. So the liver can detoxify substances like alcohol, and it can also excrete drugs like penicillin and erythromycin into bile. Now, we already talked about bilirubin excretion and bile salt synthesis. So the next function of the liver would be storage. So the liver is a large storage space for glycogen. And that's going to be the stored unit of glucose. Now, in addition to glycogen, the liver is a prime storage area for certain vitamins. So vitamins like vitamin A, B12, D, E, and K are all going to be stored in the liver. Some minerals as well. Another function is that phagocytosis. So Kupffer cells in the liver, these are going to be a very specific type of cell in the liver. Kupffer cells. These are going to be liver cells that phagocytize aged red blood cells, white blood cells, and even some bacteria. So they will phagocytize these aged and dying red blood cells, aged and dying white blood cells, and even some bacteria. The final function we'll talk about for the liver is the activation of vitamin D. So the skin, liver, and kidneys can all participate in synthesizing the active form of vitamin D, which, remember, is calcitriol. So another really important function of the liver here. Most digestion and absorption of nutrients actually occurs in a long tube called the small intestine. Now, because of this, the structure of the small intestine is really specifically adapted for these functions. So here we have this really, really long tube, this small intestine there. Now, there are three parts of the small intestine. These are the duodenum, small intestine right directly past the stomach. Then we have the ileum and the jejunum. Now, the length of the small intestine provides a really large surface area for digestion and absorption. And that large surface area is even further increased by the additional of structural features like circular folds, And then on those circular folds, there are villi. And then on those villi, there are microvilli. So really, really increasing that surface area. So circular folds are going to be folds of the mucosa and submucosa. So these are permanent ridges that begin near the proximal portion of the duodenum, and they end at about the midpoint of the ileum. So the actual order of the small intestine is the duodenum, which will go into the jejunum, which will end in the ileum. So that these circular folds will start at the duodenum and then end kind of in the midpoint. So we're going to start here at the top and then go kind of to the midpoint of the ileum here. Circular folds are along the small intestine, that whole length. Now these circular folds really enhance absorption by increasing the surface area and also causing the chyme to spiral rather than moving in a straight line as it passes through the small intestine. So it's kind of going to hit the sides, and chyme is going to kind of spiral through this entire tube, hitting as many spots as possible in the small intestine. So it's really going to increase the amount of molecules that are able to be absorbed from the food that you eat. Now, also present in the small intestine, like I said, are going to be villi. These are finger-like projections of the mucosa. And then there are microvilli, which are projections of the apical membrane of the absorptive cells. So the microvilli are very, very small, but we'll see these in the next slide. So here in the mucosal layer, you can see these really long finger-like villi. And then on each individual absorptive cell, they're going to have lots of little tiny projections, tiny little, kind of like a hair. But these are going to be tiny fingers on the absorptive cells themselves that are the microvilli. Now, the wall of the small intestine is composed of the same 4 layers that make up most of the gastrointestinal tract. So we'll see that mucosal layer with the villi. We'll see the submucosa, the muscularis, and also the serosa. So each villus, which is the singular of villi, is covered with epithelium and has a core lamina propria. Now, embedded in the connective tissue of the lamina propria, we have an arteriole. You can see the little red within the lamina propria there, but these red blood vessels go up into the villi here. So we have an arteriole colored in red. There's also going to be a venule colored in blue here. A whole capillary network between the arteriole and venule. And then also a green lacteal. Now a lacteal, this is a lymphatic capillary. So you can see that it comes from a larger lymphatic vessel here in the submucosal layer. Now, nutrients absorbed by the epithelial cells covering the villus will pass through the wall of a capillary or a lacteal to enter the blood or the lymph, respectively. And we'll talk about which nutrients will move to the blood and which ones will move to the lymph. Now, about 1 to 2 liters of intestinal juice are going to be secreted each day. So intestinal juice, this is a clear yellow fluid, and it contains a lot of water and mucus, and it's slightly alkaline. So it has a pH of about 7.6. Now, the alkaline pH of intestinal juice is really due to its high concentration of bicarbonate ions. So it has a lot of these molecules. Remember, these are bicarbonate. So this really kind of causes the intestinal juice to be a bit more basic. Now, the absorptive cells of the small intestine will synthesize several digestive enzymes, which we call brush border enzymes. So these are synthesized and secreted by the absorptive cells. And these brush border enzymes are inserted into the plasma membrane of the microvilli on the absorptive cells. So these are digestive enzymes. So here we have the wall of the duodenum, and we can see several villi here. So when we're viewing this through a light microscope, the microvilli are actually too small to be seen individually. So instead, they just form kind of this fuzzy line that we call the brush border. And this fuzzy line extends into the lumen of the small intestine. Because the microvilli greatly increase the surface area of the plasma membrane, larger amounts of digested nutrients can diffuse into the absorptive cells in a given period. Now, the brush border, which is just made up of the microvilli, this brush border also contains several brush border enzymes that have digestive functions. The two types of movements in the small intestine are segmentations and a type of peristalsis that specifically in the small intestine we call it migrating motility complexes. Sometimes we just call these MMCs. This is a specific type of peristalsis in the small intestine. So segmentations, these are going to be localized mixing contractions that occur in portions of the intestine. specifically, it's going to occur in portions of the intestine that are distended, meaning swollen due to pressure in the inside or bloated. And they're going to be distended by a large volume of chyme. So these are going to occur in distended portions of the small intestine. So distended means that they're swollen due to this large amount of chyme inside them. Now, segmentations will mix the chyme with digestive juices and also bring particles of food into contact with the mucosa for absorption. So this is just going to be kind of just a mixing type of movement. So it mixes chyme with digestive juices. And is also going to bring particles of food into contact with the mucosa for absorption. These segmentation movements do not push the intestinal contents along the gastrointestinal tract. This isn't like a forward moving type of movement. This is more of a mixing type of movement. So in contact with the mucosa for absorption. Now, after most of the meal has been absorbed, which is going to lessen the distension of the wall of the small intestine, this is when segmentation stops and peristalsis begins. So we'll put this right here. So when most has been absorbed, and the small intestine, SI, is less distended. Then segmentation ends, peristalsis begins. So again, that type of peristalsis that occurs in the small intestine is called the migrating motility complex, the MMC. This is going to begin in the lower portion of the stomach, and it pushes chyme forward along a short stretch of the small intestine before these contractions die out. So the MMC will very slowly migrate down the small intestine, really only reaching the end of the ileum in about 90 to 120 minutes. This is to cover the whole length of the small intestine. Now, after this entire length has been covered by one MMC, then another MMC begins in the stomach, and then another. And so overall, chyme remains in the stomach total for about 3 to 5 hours. Sorry, chyme remains in the small intestine, yeah. And this is just because the MMC has to move all the way down the small intestine, moving that chyme through the whole length of the small intestine before the next MMC can take the next kind of small amount of chyme and move that down the whole small intestine, and so on and so forth. Now remember, in the mouth, we have salivary amylase that converts starch into smaller molecules. So it's going to convert starches into pretty much into trisaccharides or disaccharides, into small fragments of starches. Starches are carbohydrates. Then also in the stomach, we have pepsin. Pepsin converts proteins into peptides, which are small fragments of proteins. So starch, I'll put it here, so carbohydrate. Pepsin in the stomach is going to digest proteins into smaller fragments of proteins, which are peptides. Also in the mouth, we have lingual lipase. Remember, this is going to convert fats, so fats, triglycerides, into fatty acids, monoglycerides. Essentially, it's going to digest those fats and oils. So all this to say that chyme entering into the small intestine contains partially digested carbohydrates, proteins, and lipids. We could change these fats here. These are lipids. Fats are another term for lipids, but keep everything the same here. So lingual lipase is going to digest lipids. So chyme coming in has partially digested carbohydrates from that salivary amylase, partially digested lipids from the lingual lipase, and also partially digested proteins from the pepsin in the stomach. So the completion of the digestion of carbohydrates, proteins, and lipids is going to be a collective effort of pancreatic juice, bile, and intestinal juice in the small intestine. So these are going to help to complete the digestion of carbohydrates, lipids, and proteins. This table here is a great summary of the digestive activities in the pancreas, the liver, gallbladder, and the small intestine. It does show many of the cells found in the small intestine. So we get these cells of the intestinal glands, the microvilli, the movements that happen in the muscularis layer, pretty much everything like that. It shows what these cells secrete. So we're going to see that the goblet cells and the intestinal glands secrete mucus, and it also shows pretty much the activities that they all complete. So this all pretty much is stuff that we've covered, but a nice summarizing table here. This is a super important table showing the summary of digestive enzymes. So it shows the enzymes found in saliva and also the ones that make up gastric juice. This table is going to show the enzymes that make up pancreatic juice. So the pancreas is really cool because it secretes a lot of inactive enzymes. And these enzymes are going to be later activated by other enzymes in different locations. This is really, really important because this is so that those enzymes don't start to digest the organs that secrete them. Again, the pancreas is going to secrete inactive enzymes so that the enzymes don't start to digest the organ that secretes them or start to digest the tubes that they're supposed to travel in, anything like that, so that they leave the pancreas in their inactive form, and they only become activated by other enzymes in the location that they're supposed to be acting in. Enzymes in their inactive state are often going to be named with the suffix inogen. So trypsin here, its inactive form is trypsinogen. And trypsinogen is activated by enterokinase. And enterokinase is going to be one of the brush border enzymes of the small intestine. Chymotrypsin is the active form of chymotrypsinogen. And interestingly, chymotrypsinogen is activated by trypsin. We also know that amylase is going to digest starches, also known as polysaccharides, also known as carbohydrates. We know that lipase is going to digest triglycerides. And we also know that peptidases, these are going to digest proteins. So long peptides are known as proteins. So some really important digestive enzymes here. Now here we have those brush border enzymes in the microvilli, in the plasma membrane and the small intestine. So here we can see that enterokinase that I just mentioned. So this is made by the microvilli of the small intestine, and it's going to activate trypsinogen. Remember, trypsinogen is secreted by the pancreas in its inactive form, which is trypsinogen. And once it reaches the brush border in the small intestine, enterokinase will activate it into trypsin, so that then trypsin can go and start digesting proteins, as well as activate other pancreatic enzymes that are inactive. So this specific activation occurs in the small intestine so that trypsin won't start breaking down proteins on its path from the pancreas to the small intestine. All of the chemical and mechanical phases of digestion, from the mouth through to the small intestine, are all directed towards changing food into forms that can pass through the absorptive cells lining the mucosa of the small intestine, and moving those molecules into the underlying blood and lymphatic vessels. This is going to be pretty much the end goal of digestion. So in this diagram here, this shows the first movement across a membrane. Over here, so these are the epithelial cells. These yellow cells, orangey cells, are going to be the epithelial cells of the villus. And here you can also see the microvilli, which is going to be that brush border. So you're going to see the first movement across a membrane, which is going to be this movement here. This is the transportation into the absorptive cells of the villi. And then this second movement across a membrane is from the basilar membrane of these cells to the adjacent blood capillary or lacteal. Remember, that's a lymphatic capillary. So at the top here, we'll see the purple carbohydrates first. So all carbohydrates are absorbed as monosaccharides. And the capacity of the small intestine to absorb monosaccharides is huge. So a ton of monosaccharides can be absorbed. Now, monosaccharides will pass from the lumen through the apical membrane by facilitated diffusion or by secondary active transport. And then from within the cells, all of these monosaccharides will cross into a blood capillary by facilitated diffusion. Moving on to proteins. So these are going to be these green dots here. So the green dots are going to be the amino acids because most proteins are absorbed as amino acids. These are going to be single molecules that make up proteins. Now, most are going to be absorbed as amino acids by active transport or by secondary active transport. And this is going to happen mainly in the duodenum and jejunum. Sometimes dipeptides or tripeptides can diffuse into the epithelial cells or the absorptive cells by active transport, but the most common is going to be the single amino acids. Now the smallest unit of proteins, the amino acids, will then move out of the absorptive cells by diffusion and then enter into the blood capillaries of the villus. Now, all dietary lipids, we're moving on to lipids here, which are the red colored molecules. The dietary lipids are all going to be absorbed by simple diffusion. Now, small short chain fatty acids are very hydrophobic and they contain less than 10 to 12 carbon atoms, and so they're going to be a little bit more water soluble. So these are able to dissolve in the watery intestinal chyme and pass through the absorptive cells by simple diffusion. Essentially, they're able to follow the same route that is taken by the monosaccharides and amino acids into the blood capillary. Now, last one we have to talk about here are these large short chain and the long chain fatty acids at the bottom of this figure. So besides their role in emulsification, bile salts also help to make large chain fatty acids become more soluble. So how they do that is that the bile salts in intestinal chyme will surround the long fatty acid chains and they form spheres called micelles. So this is going to be a sphere. A sphere of bile salts. Now, once formed, the micelles will move from the interior of the small intestinal lumen to the brush border of the absorptive cells. And they're going to cross into the absorptive cells just by simple diffusion. But they are only able to do this because of the micelle. So these are those triglycerides here. So these really large or long-chain fatty acids are not able to diffuse into the absorptive cells by themselves. They have to have this sphere of bile salts, this micelle, around them in order to do so. Now, once inside the absorptive cells, the long-chain fatty acids and even some of these monoglycerides, these are going to be recombined to form the triglycerides. So here we have a glyceride molecule with three fatty acids, sorry. So a single glyceride and three fatty acids makes a triglyceride. And these triglycerides are going to stick together into globules, and they're going to become coated in proteins. So they kind of make this large spherical mass inside the cell that's called a chylomicron. And the chylomicron itself is able to diffuse out of the cell into a lacteal. So chylomicrons can't travel in the blood by themselves, but they can travel within the lacteals in the lymphatic system. Now, chylomicrons are so large and bulky that they can't enter the blood capillaries. But instead, they are able to enter into the lacteals, which have much larger pores than the blood capillaries. Now, from lacteals, the chylomicrons are transported by way of these lymphatic vessels, and they're transported to the thoracic duct and enter the blood at the junction of the left internal jugular and subclavian veins. So I'm going to enter back into the bloodstream right here. Now they have this hydrophilic protein coat that surrounds each of the chylomicrons, and it keeps it suspended in blood and also prevents them from sticking to one another. Very, very important thing for them. But they're not able to enter directly into the blood capillaries because the pores are too small, but they're kind of able to enter the blood indirectly through first the lacteal and then going into the bloodstream through the thoracic duct. So let's briefly talk about the fluid balance of the digestive system in the body. So the total volume of fluid that enters the small intestine each day, which is about 9.3 liters, comes from the ingestion of liquids, about 2.3 liters, and then from various gastrointestinal secretions. So about 7 liters here from like saliva, gastric juice, bile, pancreatic juice, and intestinal juice. So this figure here is going to show us the amounts of fluid ingested and secreted on the left, and then the amounts of fluid that are absorbed on the right. So all water reabsorption is going to occur by osmosis from the lumen of the intestines. Again, it's through those absorptive cells and into the blood capillaries. The small intestine is going to do the most of the fluid absorption, about 8.3 liters. Large intestine kind of absorbing the rest to get about 9.2 liters absorbed per day, and then with a small amount being excreted in the feces. Moving on to the large intestine. So the large intestine is going to extend from the ileum of the small intestine all the way to the anus. And it's attached to the posterior abdominal wall by the mesocolon. Now remember, the mesocolon is a double layer of peritoneum. So it attaches it to the posterior abdominal wall. Now structurally, the four main regions of the large intestine are the cecum, the colon. It's going to be kind of our second region. The colon itself is going to be broken up into three parts as well. So we have the ascending colon, the transverse colon, and then the descending colon, which are all part of the colon. And then there is the rectum and the anal canal. So first part is the cecum. These are the four parts of the large intestine again. So cecum, colon, rectum, and the anal canal. Now, unlike other parts of the gastrointestinal tract, portions of the longitudinal muscles in the large intestine are going to be thickened. And it forms these three very conspicuous bands called the taenia coli, which are abbreviated here, but we can see these thickenings of the longitudinal muscle that run most of the length of the large intestine. So we can see the taenia coli here, here, and here. Now the taenia coli are separated by portions of the wall with less longitudinal muscle. Contractions of the bands will gather the colon into a series of pouches that are called haustra or haustra. And these little pouches kind of give the colon this puckered, kind of a puckered appearance. Oh, sorry, I also didn't mention there's also this sigmoid colon. It's kind of the fourth section of the colon, though it is a much smaller portion of the colon than the other three, but it is still a segment there. Now there are 5 main functions of the large intestine. First one is haustral churning. We will talk about this one yet. Peristalsis, and the movement of the intestinal contents through to the rectum. So that's going to be our first function there. Our second function is that bacteria in the large intestine will break down amino acids and proteins. As well as producing some vitamins. So some really important bacteria in the large intestine as well. The large intestine also absorbs some water, as we saw with the fluid balance slide just earlier. So large intestine does absorb some water, some ions, as well as some vitamins too. The formation of feces occurs in the large intestine. And then the last function would be defecation, the emptying of feces from the rectum. Now, the wall of the large intestine contains the typical 4 layers that we find in the rest of the gastrointestinal tract. We're going to see the serosa, the muscularis layer, which also has those longitudinal and circular layers of muscle. We'll also see the submucosa with the arterioles and venules and lymphatic vessels within that submucosal layer, and then the very large mucosal layer. Now, as you can see, the mucosal layer does look kind of similar to the mucosal layer of the small intestine, but the gastric pits are not as large, or kind of the big divots in the mucosa are not as large in the large intestine as we saw in the small intestine. Kind of have just these smaller openings to the intestinal glands instead of the pits that we saw with the small intestine. The epithelium of the large intestine contains mostly absorptive cells and goblet cells. The absorptive cells function primarily in water absorption, and the goblet cells, these secrete mucus that lubricates the passage of the contents of the colon. Now, both absorptive and goblet cells are located in the long, straight, tubular intestinal glands that extend the full thickness of the mucosal layer. Now, compared to the small intestine, the mucosa of the large intestine doesn't have as many structural adaptations that increase the surface area. There's no circular folds of the villi. However, microvilli are going to be present on the absorptive cells. So because we don't have these circular folds or these other adaptations that increase surface area in the large intestine, a lot more absorption is going to occur in the small intestine than in the large intestine. Here is just some histology of the walls of the large intestine. You can see this very large muscularis layer, smaller submucosal layer, and large mucosa layer. On the right here, we can see these openings to the intestinal glands, the intestinal gland itself, and then we can also see these large white goblet cells that secrete the mucus, and then these smaller absorptive cells that will absorb water. Movements of the colon begin when substances pass the ileocecal sphincter. So right at the beginning of the large intestine. Now, because chyme moves through the small intestine at a fairly constant rate, the time required for a meal to pass into the colon is determined by gastric emptying time. So this is the time that it takes the food to leave the stomach, to be digested enough in the stomach and to enter into the small intestine. One movement characteristic of the large intestine is called haustral churning. This top one here. So with this type of movement, the haustra remain relaxed and they become distended when they fill up. Now, when that distension reaches a certain point, the walls of the haustra contract and squeeze the contents into the next haustrum. So we can kind of think of the haustra, those little bulges in the small intestine, kind of like this. And so when some of that chyme comes through and goes into that first haustra there, it will cause that haustra to become distended. So it'll kind of look like this. And when it gets distended to a certain point, the muscles in this hostra are going to contract. So contract there, and they're going to push the chyme into the next hostra. So they're going to be moving the chyme along the large intestine that way. So that's going to be hostral churning. Now, peristalsis also occurs, although it does occur at a slower rate than we've seen previously. So it's really only about 3 to 12 contractions per minute. So it is kind of a slow movement through the large intestine. So it really happens at a much slower rate than we've seen in more proximal portions of the gastrointestinal tract. Then the final type of movement, which is mass peristalsis. This is a very strong peristaltic wave, and it begins about the middle of the transverse colon, and it very quickly drives the contents of the colon all the way to the rectum. The final stage of digestion occurs in the colon through the activities of bacteria that inhabit the lumen. Mucus is secreted by the glands of the large intestine, but no enzymes are secreted. Chyme is prepared for elimination by the action of bacteria, which will ferment any remaining carbohydrates and release hydrogen, carbon dioxide, and methane gas. Bacterial products that are absorbed in the colon include several vitamins that are needed for normal metabolism, so including some B vitamins and vitamin K. By the time that chyme has remained in the large intestine for about 3 to 10 hours, it has really become solid or semi-solid because of water absorption, and at this point it's called feces. Even though like 90% of water absorption occurs in the small intestine, the large intestine absorbs enough water to make it an important organ in maintaining the body's water balance. So about 90% of water absorption occurs in the small intestine. Mass peristaltic movements will push fecal material from the sigmoid colon into the rectum. Now, the resulting distension of the rectal wall will stimulate the stretch receptors, which initiates the defecation reflex. And this defecation reflex will result in defecation. This is going to be the elimination of feces from the rectum through the anus. So, in response to the distension of the rectal wall, the receptors, the stretch receptors, are going to send their sensory nerve impulses to the sacral spinal cord. Now, the motor impulses from the cord will travel along parasympathetic nerves back to the descending colon, to the sigmoid colon, rectum, and the anus. And the resulting contraction of the longitudinal rectal muscles will shorten the rectum and increase the pressure within it. Now, this pressure along with the voluntary contractions of the diaphragm and abdominal muscles, plus the parasympathetic stimulation, is going to open the internal anal sphincter. The external anal sphincter is voluntarily controlled. This table here summarizes the digestive activities in the large intestine. It shows the different structures, the lumen, mucosa, and muscularis, shows the activity with them, each of them, and then their functions as well. And then this table summarizes the functions of all the digestive system organs, all those accessory organs, and the gastrointestinal tract organs. Also going to show their functions here, so good, just summarizing table here. Now, digestive activities will occur in 3 overlapping phases. These are the cephalic phase, the gastric phase, and the intestinal phase. So, first, during the cephalic phase of digestion, this is where things like the smell, the sight, even like the thought or initial taste of food is going to activate neural centers in the cerebral cortex, the hypothalamus, and the brainstem. Really, the purpose of the cephalic phase of digestion is to prepare the mouth and stomach for the food that's about to be eaten. Now, once food reaches the stomach, this is when the gastric phase of digestion begins. We have these neural and hormonal mechanisms that will regulate the gastric phase of digestion, and these both work to promote gastric secretion and gastric mobility. Then the intestinal phase of digestion begins once food enters the small intestine. So this is when it reaches the stomach in the gastric phase. And then the intestinal phase is once it's reached the small intestine. So in contrast to the reflexes initiated during the cephalic and gastric phases, which will stimulate the stomach secretory activity and motility, those occurring during the intestinal phase have inhibitory effects that will slow the exit of chyme from the stomach. So this intestinal phase helps to prevent the duodenum from being overloaded with more chyme than it can handle. So we're going to kind of slow down digestion in this phase. So here we have a negative feedback diagram that shows the regulation of pH of gastric juice as well as gastric motility during the gastric phase of digestion. So if we have an increase in the pH of the gastric juice, or also distension of the stomach walls, this is detected by chemoreceptors and stretch receptors in the stomach, and it is sent to the control center, which is in the submucosal plexus. Remember that this is a plexus in the enteric nervous system, the brain of the gut. So the submucosal plexus will then send parasympathetic nerve impulses to the effectors, being the parietal cells and the smooth muscle in the stomach wall. Now this will cause the parietal cells to secrete hydrochloric acid to decrease the pH or make the stomach acid more acidic. And this will also cause the smooth muscle to contract more vigorously in the stomach. So this will increase the acidity of the stomach chyme and also mix the stomach more rapidly. This will return us back to homeostasis, having a normal blood pH, or a normal, stomach acid pH, and also reduce the stretching of the stomach walls by emptying the contents into the small intestine. This table here summarizes the major hormones that control digestion. So these are some pretty important hormones here. So we're looking at gastrin, secretin, and CCK. So it'll show what they stimulate as well as where they are secreted from, and gives their major and minor effects here. So just like every other system of the body, aging has an effect on the digestive system. So it's going to result in decreased secretions and also decreased motility of the digestive system. With aging comes a decrease in muscular strength of all the muscular tissue types. So we're going to get a loss of strength and tone in the muscles of the digestive system. We'll also get changes in the neurosecretory feedback and then also a decreased response to pain and internal sensations. So these are going to be pain of the gastrointestinal tract or pain of the accessory organs of the digestive system, that internal pain and internal sensations. And then here we have our homeostasis slide, the contributions of the digestive system to all the other body systems. So the digestive system essentially is going to get all the nutrients from what we ingest, what we eat, and it will send those nutrients to all other systems of the body. Glucose, amino acids, lipids, carbohydrates, all of these building blocks for every other system of the body is mostly going to come from what we eat. And so these are going to be broken down by the digestive system and sent to all the other systems of the body. As well as water. Water is a huge, huge resource that almost every other system of the body needs. All of our cells need water. When we are dehydrated, it can really wreak havoc on all of our body systems as well as just the cells in the body systems. So the digestive system is really important for that intake of water as well. So give a quick look over this slide for the different systems of how the digestive system affects all of them. And that will be the end of chapter 23.